Hello students, welcome to this session. Let's study carbohydrates.
Hello students, today we will see now our macronutrients, that is nutrients required in large quantity. They are first is carbohydrates. So carbohydrates definition, they are hydrates of the carbon, hydrogen and oxygen group. So sources are like plants, leaves and all, and fruits plus wheat, rice, potato. So classification of carbohydrates is of mainly three types, that is monosaccharide, disaccharide and polysaccharide. So monosaccharide means they are simple sugars. One sugar molecule is there. Example is glucose, fructose. Disaccharides, they contain two sugar molecules. Example is sucrose, lactose, maltose. So lactose present in milk and sucrose present in sugar and maltose. Third type is polysaccharides. So they are more than 10 molecules of monosaccharides, that is simple sugars present in polysaccharide. So they are of two types, homopolysaccharide and heteropolysaccharide. So if the sugar molecules are same, that is monosaccharide, they are homopolysaccharide, and examples are starch, cellulose, glycogen, dextrin. And if the monosaccharides are of different types, then they are called as heteropolysaccharide, and example is mucopolysaccharide. Now digestion of carbohydrates usually it takes place in digestive system. So digestion of carbohydrates starts from the chewing mechanism, that is from the salivary gland which secretes saliva, which contains salivary amylase, and then that breaks down cooked starch into the maltose, that is disaccharide. Then in pancreas, pancreatic juice is present, and which contains pancreatic amylase, and which breaks down again the remaining part of the starch into maltose, that is polysaccharide is converted into disaccharide. Then in small intestine, the intestinal juice is secreted, and then there are two enzymes, invertase and maltase. So invertase breaks down sucrose, that is disaccharide, into monosaccharide, that is glucose, and maltase breaks down maltose into glucose, and then finally this monosaccharide, that is glucose, simple sugar is formed and which is absorbed into the blood. Now we will see importance of carbohydrates. So carbohydrates are easily available and least expensive source of energy for the body. 1 gram of carbohydrate on oxidation gives 4 kilocalorie of the energy, and glucose is a main source of the energy. Glucose is converted into glycogen and stored in liver and muscles, and when the fasting state or when we do not take food, that time the glycogen is again converted into glucose and then maintain the blood sugar level. Muscles glycogen supply energy to muscles, and some part of glucose is converted to fat for energy reserve, that is in fats. And then because of this the roughage diet of the polysaccharides, there is no constipation and there is no gastrointestinal disorders. To maintain the bowel movements, this rough fiber food is important for the digestion. Thank you.
Hello students, welcome to this session. Let's study about lipids.
Hello students, today we will talk about lipids, that is fats. So they are organic compounds of combination of fatty acid plus glycerol. So classification is simple lipid, compound lipid and derived lipid. So simple lipid, they are esters of fatty acid plus glycerol. Example is fats, which is present in tripalmitin, coconut fat, animal fat, or wax, which is vitamin D esters or true wax. Then derived lipids, they are found in breakdown of the simple or compound lipids. So second type is compound or conjugated lipids. So they are esters of fatty acid plus glycerol plus other compound. So like phosphoric acid is present, then it is called as phospholipid. So they are present in nervous tissue, brain, liver or kidney. An example, they are cephalin, cardiolipin, plasmalogen. So they form a structure like mitochondria, or cephalin helps in coagulation, or they act as a prosthetic group to enzymes, or sometimes cardiolipin present in mitochondria. Then fatty acid plus glycerol plus carbohydrate, if it is present, it is called as glycolipid. Example is cerebrosides and gangliosides. If lipid is present in combination with fatty acid plus glycerol, it is lipoproteins. So they are HDL, LDL and VLDL, that is high density lipoprotein, low density lipoprotein and very low density lipoprotein. So importance of lipoproteins, they are present in cell membrane which maintains the structure of cell membrane. Then they help in a transport of lipids to tissues, and they maintain the structure of mitochondria, microsomes. Then if the combination of fatty acid plus glycerol plus sulfuric acid is present, it is called as sulfolipids. Then fatty acids, they are found in hydrolysis of lipids. So they can be saturated fatty acid, unsaturated fatty acid, or essential fatty acid, or steroids. So saturated fatty acids, it has the hydrogen atoms attached to each carbon atom with a single bond. They are solid at room temperature. So examples are acetic acid, butyric acid, palmitic acid, stearic acid. Unsaturated fatty acid, the hydrogen atom missing, it contains double bond. They are oils at room temperature. So oleic acid, linoleic acid, linoleic acid, arachidonic acid. Essential fatty acid are polyunsaturated fatty acid which are not synthesized in body, have to be provided in the diet. So they are linoleic acid, linoleic acid, and arachidonic acid. Steroids, they are nucleus of 3 to 6 membered carbon rings and 1 to 5 membered carbon ring. So importance of steroids, they are naturally present, that is male sex hormones, testosterone, or female sex hormones, estrogen and progesterone, or they can be present in corticosteroids like dexamethasone. Then importance, 1 gram fat gives 9 kilocalorie of the energy. They act as a insulators and protect organs or body. So they are present around the organs and they help the protection of organs, or they are present in fatty tissue, and because of that they act as a shock absorber for our body. And then they store the fats, that is it is a form of energy. And then they are important component of the cell membrane. They form hormones. Steroids, they are component of nervous tissue, mitochondria, or the vitamin A, D, E, K, they are fat soluble. The essential fatty acids are polyunsaturated fatty acid, and they have to be provided in diet, that is linoleic and arachidonic acid. So this is the importance of the lipids. So today we will talk about proteins. So proteins are polymers of amino acids and they contain carbon, hydrogen, oxygen and nitrogen. So they are also called as polypeptides. So amino acids combine with a peptide bond and many peptides form the polypeptide. So actually they are of 3-4 structures like primary, secondary, tertiary and quaternary. So polypeptide chains are there and all these chains together form the quaternary structure, that is proteins. Classification, it is simple protein, derived protein and conjugated protein. So simple protein means it is only protein. So examples, albumin, globulin, or keratin which is present in nail, or collagen which is present in skin and bone, or elastin which is present in ligaments. Derived proteins, they are breakdown products of the proteins in while digestion. The conjugated proteins, they are combination of simple protein plus non-protein substance, and that non-protein substance is called as prosthetic group. So a few examples like phosphoprotein, it is a combination of protein plus phosphorus. Example, milk. So in that casein is there, or egg yolk, in that vitelline is there. Lipoprotein is a combination of protein plus lipid. Example, egg yolk. Glycoprotein is a combination of protein plus sugar. Example, mucin. Nucleoprotein, protein plus nucleic acid. So example, DNA, RNA. Porphyrinoprotein is a combination of protein plus metal, that is porphyrin group and metal. So example is hemoglobin, in which iron is there as a prosthetic group. Classification of proteins based on amino acids can be complete, partially complete and incomplete. So complete proteins when it contains all essential amino acids to maintain normal life and growth. Partially complete when few amino acids are missing, and then it maintains life but not the growth. And incomplete proteins means essential amino acids are lacking and which do not maintain life and growth. So they are incomplete proteins. Functions, they are nutrition, provide, then transport of lipids, osmotic water balance, buffer action, and blood coagulation takes place because of the fibrinogen and it prevents blood loss. Then respiratory function by hemoglobin, that is transfer of oxygen to tissues and transfer of carbon dioxide from tissues to lungs, sorry, from tissues to outside. Digestion takes place in stomach. So in that gastric juice is there which contains pepsin and hydrochloric acid. So this pepsin breaks down proteins into polypeptide plus peptide. Then pancreas contain pancreatic juice which contains trypsin, and it breaks down polypeptides or peptodes into amino acids. Small intestine contains intestinal juice which contains erepsin, and that breaks down polypeptides or remaining part of the peptones into amino acids. And finally, simple form, that is amino acid, is formed from the proteins, and then that is absorbed into the blood. Importance: they are building blocks of the body. They are essential for growth and wear and tear of the body. They are present in bones, tooth, skin, nails, hair, blood cells. They are source of energy, and also they are present in DNA and RNA, which is present in nucleus and cytoplasm. Thank you. Hello students, today we will talk about amino acids. So they are proteins are made up of units. That units are called as amino acids. So amino acid is a monomer of the polymer, that is protein. So classification: they are essential amino acids, semi-essential amino acids, and non-essential amino acids. So essential amino acids are those amino acids which cannot be synthesized in the body, and you have to take it in a diet. Example: methionine, threonine, tryptophan, valine, isoleucine. Semi-essential amino acids are those amino acids which can be synthesized from other amino acids like cysteine, tyrosine. And non-essential amino acids are those amino acids which can be synthesized in body and no need to take in a diet. So example: glycine, alanine, cysteine, aspartic acid, glutamic acid, proline, hydroxyproline are all non-essential amino acids. Or other classification based on the nature of the amino acid: they are neutral, sulfur amino acids, acidic, basic, and aromatic amino acids. So neutral, glycine is an example. Sulfur amino acid, they are cysteine, methionine. Acidic amino acid, they are aspartic acid, glutamic acid. Basic amino acid, they are alanine, and aromatic amino acid, they are tyrosine. Importance: so amino acids are supplied in diet to maintain normal growth and nitrogen balance and health. Aspartic acid, glutamic acid are involved in transmission of impulses in the nervous system. So they are mainly for the nerve impulses. And many hormones are amino acids like adrenaline, then thyroxine, that is T3, T4, secreted from the thyroid gland. Glycine also required for the synthesis of the heme. Or amino acids required for the synthesis of the nucleotides. They are the part of the DNA and RNA. And abnormalities in transport of amino acids may result in disease. So amino acids mainly required for the formation of the proteins. Thank you. Hello students, today we will talk about enzymes. So what are enzymes? They are synthesized by cells of all living organisms. So they are biocatalysts. They accelerate the multitude of metabolic reactions upon which life depends. So all metabolic activities are accelerated because of the enzymes, and they are proteins in nature, and their catalytic activity depends on the structure of the polypeptide chains. So for example, substrate is converted into product by the enzymes. So enzyme specificity is a property, that is particular enzymes act only on particular chemical group. Factors affect the enzyme's activity are substrate concentration, enzyme concentration, then temperature, that is 37 degrees Celsius, pH it should be between 5 to 9, then ionic strength, oxidation reduction potential. Now classification. So they are classified according to the different functions. So first is oxidoreductases. So they catalyze oxidation reduction reaction which gives energy. So example of that is alcohol dehydrogenase. Second group is transferase. So they carry out transfer reaction in which one group is transferred to another. So examples are AGOT, AGPT. So serum glutamate oxaloacetate transferase or serum glutamate pyruvate transferase. Hydrolases, they catalyze hydrolytic reaction. Examples are amylase, lipase. These are the pancreatic enzymes. Lyses, so they catalyze reaction resulting in lysis, that is elimination of group. So example is maltase, lactase, sucrase. So these enzymes break down disaccharide into monosaccharide. Then isomerases, so they catalyze isomerization reaction which bring about intramolecular rearrangement in molecule. So example is L-alkyline isomerase. Ligases, so they catalyze joining reaction which link two molecules. So they bind two molecules, so they are called as ligases. Other classification can be it is functional enzymes, so they are present in all the times and perform physiological functions in the blood. Example, in blood coagulation enzymes like thrombokinase, then lipoprotein lipase. Non-functional enzymes perform no physiological functions in blood. The enzymes of exocrine secretion, intracellular enzymes are non-functional enzymes. Uses of enzyme is for the assessment of the severity of the organ damage, then follow trend of the disease, that is follow up, then differentiate particular type of disease and determine post operative risk. So example, amylase is used in the conditions, if it is raised, it can be acute pancreatitis, pancreatic disease like pancreatic cancer. AGPT, SGOT increased in infective hepatitis. CPK, MB, SGOT, LDH, these enzymes are increased in myocardial infarction. Alkaline phosphatase increase in bone disease like rickets or obstructive jaundice. Acid phosphatase increase in prostatic cancer. Lipase similar to amylase, it is increase in pancreatic disorder like pancreatitis, alcoholic pancreatitis, viral pancreatitis, pancreatic tumor.
Secondly, CPKM. So CPK is creatinine phosphokinase. हे टोटल तीन प्रकारचे असतात, CPK-MM, CPK-DB and CPK-ME. So CPK-MM is increase in muscular disorders, CPK-DB is increase in brain disorders, and CPK-ME is increase in heart disorders, that is myocardial infarction. And third enzyme is LDH, lactate dehydrogenase. So it is increased, then it is also seen in cardiac disease like myocardial infarction. Now we will see pancreatic enzymes. So there are two, amylase and lipase. So amylase and lipase. Amylase is generally required for the digestion of the carbohydrates and lipase is generally required for the digestion or breakdown of the lipids. If they are increased, then it is seen in pancreatitis. Pancreatitis can be because of the viral pancreatitis, alcoholic pancreatitis or pancreatic tumors. Thank you.
Let's study vitamins and minerals. Vitamins, these are the micronutrients important of the diet. So there are two types, fat soluble and water soluble. So fat soluble are A, D, E, K, and water soluble are B complex and vitamin C. So A is source vegetable, carrot, tomato, fish, milk. Function, growth, vision at night. Deficiency, poor growth, night blindness, dryness of eyes. D, sunlight, milk, fish, egg. Function, calcium and phosphorus absorption. Teeth and bone formation. Deficiency, rickets in children. Osteomalacia in adult, badly formed teeth. E, milk, fish, egg and meat. Function, non-reproduction. K, source, leafy vegetables, cabbage, eggs. Function, blood clotting. Deficiency, gums and skin bleeding, increased clotting time. Vitamin B1, that is thiamine, source, milk, nuts, meat, cheese, peas, cereals, vegetables. Coenzyme, nerve function. Deficiency, beriberi, loss of appetite and digestion, polyneuritis, decreased nerve function. Vitamin B2, that is riboflavin, milk, nuts, meat, cheese, peas, cereals. Coenzyme, tongue infection, skin infection, eye infection. B6 pyridoxine helps in RBC formation, deficiency, nervous disorder. B12 cyanocobalamin, function RBC maturation and growth, deficiency, megaloblastic anemia. Folic acid, leafy vegetables, RBC maturation and growth, deficiency, megaloblastic anemia. Vitamin C, citrus fruits, cabbage, vegetables, tomatoes, the function, teeth and gum bones, deficiency, bleeding gums, swollen gums, joint pain, scurvy. Minerals, first calcium, normal range 8.4 to 10.4, function, normal function of body, important constitute in bone and teeth, nerve function, muscle contraction, blood clotting. If decrease, then rickets, osteoporosis, osteomalacia, tetany, increase clotting time. If increase, calculus in gallbladder or kidney. Phosphorus 2.5 to 4.5 in adults and child 4 to 6 mg/cent. Function, important constituent of bone in formation of ATP, adenosine triphosphate. Decrease in osteoporosis. Third, sodium 135 to 155 millivolt per liter. Function extracellular fluid control, acid base balance, nerve function, muscle function. Decrease hyponatremia, seen in diarrhea, vomiting, dehydration. Increase hypernatremia. Potassium 3.5 to 5.5. Function intracellular fluid control, acid base balance. Decrease hypokalemia, seen in dehydration. Increase hyperkalemia. Fifth is iodine, growth and in the field of body. Thyroid gland requires iodine for synthesis of thyroid hormones like T3 and T4. Decrease low iodine, low T3 for hypothyroidism. Sixth, iron function hemoglobin formation stored in liver. Decrease iron deficiency anemia, increase hemosiderosis.
Hello students, welcome to this session. Today we are going to study urine examination. Hello students, today we will see bilirubin metabolism. So bilirubin metabolism is for the formation of jaundice, and jaundice is a clinical symptom of the liver disease, and that is yellowish discoloration of the skin or sclera or mucous membrane. So in short, bilirubin metabolism starts from the hemoglobin, RBC destruction, then hemoglobin is broken down into globin plus heme, and then heme is broken down into iron plus biliverdin or protoporphyrin, and that is called unconjugated bilirubin. Then that enters the liver, and then conjugation takes place with glucuronic acid in the liver, and then conjugated bilirubin is formed, and then that bilirubin through the blood circulation it goes to gastrointestinal tract or to the kidney. So in the gastrointestinal tract, in the large intestine, by the action of the bacterial flora, it is converted into the stercobilin, and then it is excreted into the feces, and that gives brown color or yellowish color to the stool. And then in kidney it is excreted as urobilin or urobilinogen, and that gives yellow color to the so this is bilirubin metabolism. Hello students, today we will talk about classification of jaundice. So what is jaundice? It is yellowish discoloration of the skin, sclera or mucous membrane, and in that bilirubin is increased, so it is also called as hyperbilirubinemia. So there are three types of jaundice: hemolytic jaundice, hepatic jaundice or hepatogenous jaundice, and obstructive jaundice. So these hemolytic and hepatogenous jaundice are also called as medical jaundice, and obstructive jaundice is called as surgical jaundice because their surgery is the treatment. So now first we will see hemolytic jaundice. So this hemolytic jaundice is because of the excessive destruction of the RBCs, and because of that the hemoglobin is released. It is broken down into heme and globin, and then heme is again broken down into iron plus porphyrin, and then that porphyrin is unconjugated bilirubin. It is also called as biliverdin, and then because of that in hemolytic jaundice there is increased unconjugated bilirubinemia. So total bilirubin is increased and indirect bilirubin is increased, that is unconjugated bilirubin. And this hemolysis the excessively maybe because of the hemolytic disease of newborn, or hemolysis because of the mismatched blood transfusion also the reason. Then second is hepatogenous jaundice or hepatic jaundice. So it is due to damage of the liver cells, maybe up to the necrosis of the liver cells, and in that the conjugating and excretory function of the liver is affected. So examples of hepatic jaundice are viral hepatitis, alcoholic hepatitis, or maybe cirrhosis, that is fibrosis of the liver. So in that conjugated bilirubin is increased, total bilirubin is increased, and then liver enzymes are also increased because of the damage to the liver cells. Third is the obstructive jaundice. So this obstruction is because of the either stone or fibrosis or tumor in the biliary tree, that is either in the bile duct, common bile duct, gallbladder, or where the common bile duct enters into the duodenum. So that site also may be obstruction, and because of that the bile does not enter into the small intestine, that is duodenum, and because of that there is no digestion and absorption of fat. So fat is excreted in stool as it is, and because of that the color of the stool is white or clay color, and itching all over the body is the main symptom. So in jaundice main symptoms are abdominal pain, nausea, vomiting. In hepatic jaundice there may be hepatomegaly. So these are the basic symptoms, and then in that urine will be the dark yellow in color in hepatic and obstructive jaundice, and urine bile salt bile pigment are present. So now we will see investigations in different types of jaundice. So urine bile salt and bile pigment is increased in hepatic and post-hepatic jaundice. It is normal in pre-hepatic. Urine urobilinogen is increased significantly in pre-hepatic, that is hemolytic jaundice, and normal in hepatic and post-hepatic jaundice. So total bilirubin is increased in all these three types of jaundice, but direct is increase in hepatic and post-hepatic, and indirect, that is unconjugated bilirubin, is increase in hemolytic jaundice. HGPT and HGOT, these two liver enzymes are increased in hepatic markedly, and in post-hepatic also they are mild increase. Alkaline phosphatase, it is markedly increasing post-hepatic jaundice, that is obstructive jaundice. Total protein, albumin globulin ratio, these are initially they are normal, but maybe abnormal in later stages of the jaundice. Thank you. Hello students, today we will see about LFT, that is liver function test. So indications are to find out different types of jaundice like hepatic, pre-hepatic or post-hepatic jaundice, because treatment is different. Then to assess severity of the liver damage, whether hepatitis or cirrhosis is there, to follow up after the treatment. Then in infective hepatitis we can do screening, or if hepatotoxic materials are there in industry, to see that, or to see the effect of hepatotoxic drugs to treat other disease like in tuberculosis, INH, rifampicin is used, they are hepatotoxic. So for that we do LFT. So LFT is number of investigations done to determine the liver function, that is the severity of the liver disease. So there are different tests done. So first group 1 test, in that bile pigment and bile salt excretion is seen. So that is in urine we can see bile salt bile pigment. So bile salt by Hayes test and bile pigment by Faucett test. Then in blood serum bilirubin, that is total, direct, indirect, we can see, and then we can see which type of jaundice is there. Means jaundice is there or not there, and then which type of jaundice is there. Then group 2 test, they are liver enzymes, so to see the severity of the liver disease. So AGPT, AGOT, alkaline phosphatase, gamma GT, that is gamma glutaril transferase, these tests are done. Then group 3 test, changes in plasma proteins are these investigations are done to see the severity of the liver damage. So total proteins, albumin globulin and albumin globulin ratio. And then group 4 contains other tests like bile acid, prealbumin or antitrypsin, hepatoglobin, prothrombin time, cholesterol, triglyceride, ammonia, liver biopsies. These are different tests done to see different liver diseases and damage in the liver. Thank you. Hello students, welcome to this session. Today we are going to study urine examination. Hello students, today we will see renal function test. So renal function test is the investigations done to see the function of the renal, that is kidney. So these function tests are divided into 3 types. So that can be because of the pre-renal, renal or post-renal. So pre-renal means other than urinary system. Then renal means it is because of the kidney involvement, and post-renal means it is because of the either ureter, bladder or urethra. So now pre-renal condition means it can be dehydration, or it can be severe bleeding or shock or cardiac failure or pregnancy. So in that you can see altered renal function. Then in that you will see blood urea, nitrogen or creatinine, they are normal. Then renal causes may be because of the changes in renal anatomy and physiology, that is it can be nephritis, glomerulonephritis, nephrotic syndrome, pyelonephritis, urinary tract infection. So in that renal functions are altered, that is urea, creatinine are increased. Then post-renal causes, it can be because of the obstruction to urine flow, because of the enlargement of the prostate or stone in urinary tract, that is either ureter or bladder, or tumor in urinary bladder, cystitis, urethritis. So all these condition, in that urea and nitrogen will be absent, and then you can see the obstruction in the post renal pathway by sonography. So these are the renal function test, the causes we can find out. Then what are the indications of renal function test? So patient with history of renal disease, that is, in that symptoms are early morning swelling around the eyes, mostly on face, and swelling all over the body, or if a person is standing, then swelling over the legs, or ankle edema is there. Then history of urinary tract infection, that is either burning urination, or itching is there, or the frequency is increased, or the painful urination, or history of urinary stone, so you will get pain over the abdomen at the lumbar region. Then pregnancy with proteinuria or nephrotic syndrome, the proteinuria 4 plus, or to differentiate pre-renal, renal, post-renal causes. Then if renal condition is there, then to know the severity of the renal disease or severity of the renal failure, or if patient with diabetes, hypertension, alcoholism, then in that conditions you have to do all renal function test, that is renal kidney investigations. Thank you. Hello students, today we will see kidney function test. So kidney function test, KFT or RFT, they are tests done to know the kidney disease severity. So these are the investigation to know the functions of the kidney. So there are groups. So first, group 1 is to differentiate pre-renal from renal and post-renal conditions. So in that, blood urea nitrogen, serum creatinine, routine urine examination, that is physical, chemical and microscopic examination is done to know the whether kidney is involved or not involved. If these are normal, then it can be because of the pre-renal or post-renal causes. If blood urea nitrogen, serum creatinine and routine urine examination shows abnormality, then it is renal involvement. Then group 2, to assess the severity of the renal disease. So now if renal involvement is there, then you have to know the severity of the disease. So for that we have to do total protein, albumin globulin ratio. So in that, total protein is decreased, albumin is decreased, and in urine you will have proteinuria, that is 4 plus. Then quantitative determination, that is 24 hours urine proteins can be done, or electrophoretic fractionation of serum proteins can be done. Then serum cholesterol. So here you can, the disease can be either nephritis, glomerulonephritis, pyelonephritis, urinary tract infection, nephrotic syndrome, or renal failure. And then group 3 test to measure the glomerular filtration. So this is mainly done in renal failure. So they are creatinine clearance test or urea clearance test. So these are the different investigations done to know the functions of the kidney, whether they are normal or abnormal. Thank you. Hello students, welcome to this session. Today we are going to study urine examination. Hello students, today we will see about thyroid gland. So normally it is present in neck, and two hormones are there secreted by thyroid gland. They are T3 and T4, triiodothyronine and tetraiodothyronine. So functions of thyroid and hormones mainly for the normal metabolic activity, physical development and mental development. And TSH, it is thyroid stimulating hormone secreted by anterior pituitary gland, and which stimulates the thyroid gland to secrete T3 T4. So mainly two diseases are there in thyroid gland disease. So it can be hypothyroidism, so when the thyroid hormones are low. So mainly symptoms are that enlargement of the thyroid gland, it is also called as goiter, or abnormal weight gain, low metabolic activity. In adults it is called as myxedema, in infants it is called as cretinism, and in cretinism mainly child shows big swollen protruded tongue, and then low mental activity and low physical activity. So second is hyperthyroidism, in which thyroid hormones are increased, and symptoms can be enlargement of the thyroid gland, weight loss, nervousness, fatigability, excessive sweating, and sometimes protrusion of the eyeballs also seen, that is called as exophthalmos, and increased body temperature and heart rate can be seen. Thank you. Hello students, now we will see indications of thyroid function test. So sudden weight gain, sudden weight loss, heat intolerance to either cold intolerance, or infertility, long-term constipation or diarrhea, delayed development in the children, or goiter, that is thyroid gland enlargement can be seen from outside, or difficulty in breathing or chewing we can see, or exophthalmos, that is protruded eyeballs, or tremors. So these are all the indications for the thyroid function test. So there are two panels, panel 1 and panel 2. So in panel 1, routine investigations like serum T3, T4 and TSH is done, and in panel 2, free T3, free T4, ultra sensitive TSH, and then other investigations like antibodies can be done to know the autoimmune status. The methods are either RIA, ELISA or chemiluminescence. And if T3, T4 are elevated, it is hyperthyroidism. If T3, T4 are decreased, it is hypothyroidism. Thank you.
Hope you find this session useful for your understanding and for your exams. Thank you.